Hello everyone, my name is Taksh Bhatia, and today I'm going to be presenting about the possibility of utilizing CRISPR-Cas9 for curing HIV, or the human immunodeficiency virus. So here's just my table of contents right here. So as I mentioned, the title of my paper is If CRISPR-Cas9 Can Cure HIV. So just to give a bit of background information, HIV stands for the human immunodeficiency virus. It's a virus that specifically attacks the immune system of humans specifically. And those cells that are affected by HIV are called CD4 plus T cells. And these cells can be found all over the body. But the issue is, when HIV does attack these CD4 plus T cells, they tend to die off rapidly. And because of this rapid death, due to it being so hard to regenerate these cells, it can immensely devastate the human immune system. And when humans have less immune cells, this puts them at risk to contract other diseases specifically. And for example, what might be a cold, for example, rather than just giving you some symptoms of a runny nose or some sneezes, for example, it can turn into a life-threatening disease. So it's really important that people have greater knowledge about this disease and start treatment immediately. Now, the issue is, with HIV, there's no cure. There are some treatments such as ART, which is antiretroviral therapy, and in this specific form of therapy, pills are taken, for example, or maybe monthly or bi-monthly injections. But this is a regimen that must be done very strictly, as if it's not maintained, what will happen is it will give the virus time to build resistance against this antiretroviral therapy. And if it does form resistance, these levels of HIV, rather than being controlled at a very low level, they can start to spike again and cause greater devastation to the immune system. And due to this sort of therapy having to be required for the rest of one's life after HIV is diagnosed and treatment is begun, it can become an incredibly difficult part to maintain throughout one's life. And so, with this sort of solution of CRISPR as serving as a once and for all potentially treatment for removing HIV genetic material from the human body, it's possible that people who have HIV or potentially at risk at or want to prevent themselves from it, they can undergo this specific form of gene therapy to prevent any form of infection from HIV in the future. So my research questions are, how does HIV infect the immune system's CD4 plus T cells? So HIV utilizes a specific receptor called the CD4 receptor. And upon the CD4 receptor, it also requires to bind to one other co-receptor. This can be either the CCR5 co-receptor or the CXCR4 co-receptor. If HIV is unable to bind to this receptor and one of the co-receptors, it is not able to infect the cell or inject its DNA or its genetic material into the cell. My next question was, how can we prevent HIV from binding to cells? This will be addressed later in the presentation. And afterwards, how can we edit the T cell genome to terminate gene expression for a specific receptor? And the question is, would it cause complete fortification from HIV? Now, another question, of course, is how could we transport this gene editing technology? As there are some concerns. For example, if we are introducing completely foreign technology into a human cell, there's going to be a natural reaction to that specific entering, and it could cause potentially some pretty bad side effects, and it's important that we address those. These questions are really important because when you put them together, it can really form an effective system utilizing gene therapy to cure an incredibly devastating disease. In the 1980s and 90s, for example, HIV was incredibly devastating amongst communities such as the LGBTQ plus community. And even to this day, there are 39 million cases globally, 26 million of which are in low socioeconomically successful areas such as Africa. And if we can potentially make this a more affordable solution, it can be absolutely groundbreaking for many of these communities. The objective of this research is to provide an effective solution to remove HIV and protect uninfected individuals from it. And as I mentioned before, it can be a very, very effective alternative than strict regimen treatments like ART, because once I said, like I said before, this is more of a one-time treatment rather than a continuous treatment for the rest of one's life. So here are some past or current, at least, studies being done on this sort of related field. So one method that some researchers have used are lentiviral vectors. What these lentiviral vectors do is it's sort of like a container of sorts. One can think of as sort of a container. The biological term, this could be sort of like similar to a vesicle, for example. And what these sort of like lentiviral vectors do is they carry plasmids with transgenes specifically. And these transgenes, what they do is into a specific DNA segment of interest, for example, if we are to edit that, it'll insert those transgenes into that specific segment of DNA to cause a mutation. It could be a frame shift mutation. It could be a sort of mutation that prevents that gene from expressing or changes the form of expression. And these sort of lentiviral vectors have been used for lots of current gene editing specifically in the past and even today. One study, for example, that relates to HIV is SIV, which is the simian immunodeficiency virus. This is, it's like the sister virus of HIV, and it's present in many of human relatives, such as macaques, chimpanzees, and those many of our closest relatives in the animal world. And this study focused on how we could use these vectors to treat SIV. And studies have shown that these treatments of using transgenes have almost up to 100% efficiency in reducing capsid production of HIV in these cells. For example, because many of these T cells, because what HIV does is when it injects its genetic material into the T cells, those T cells are used as factories to produce more capsids, more HIV capsids that spread across the human body. And so that sort of method was utilized here as well to prevent the protein capsid production, so that way less HIV viruses are produced that can infect other cells. And this specific transgene treatment, almost 100% efficiency in bringing that treatment down to zero, which is incredible to say the least. Now, there are some issues to that. For example, with lentiviral vectors, an issue that can arise is because it's more of a long-term thing, it can cause random insertions, for example, random genes to stop working, or random insertions where it just goes into the wrong gene itself. There's a lot of room for error in that. And if we do take this into account, which we should, of course, it can have long-term effects, such as causing other important genes that need to be expressed to not be expressed. And due to that, can cause some very adverse symptoms. And it's important that we address this aspect of safety in gene editing. Another one is electroporation. So electroporation, the way it works is that, for example, if we were to do it in a cell culture, for example, electrodes would be used to transmit an electrical current through those cells. And those cells, because of the electric current, what happens is its cell membrane, or at least the cell membranes, they become more porous, like tiny, tiny holes, like pores, for example, they appear. And this allows for easier transport of extracellular objects. And what makes this really appealing is that it's a lot more affordable. It's been used for the past couple decades on a lot of other experiments and other fields in science. And due to this, it really addresses a key aspect and a key concern of gene editing, which is affordability, because lots of this gene editing technology is hard to access in these low socioeconomically successful areas such as Africa. And with electroporation, because it provides an affordable method, if implemented, it can really bring down the cost of specific gene editing processes. However, there are some drawbacks to it, as there were for lentiviral vectors. For example, with electroporation, in vivo trials, which are more on, for example, living beings such as humans, there have been tests done on humans with electroporation. And a specific drawback that's been identified is that with electroporation, when the electrodes are placed on the skin, it can cause severe symptoms. For example, skin edema has been a potential side effect caused by electroporation. Another one is due to the electric shocks, muscle mass and muscle strength. They have gone down to 40%. And it does take time for those muscles to regenerate and regain that strength that it was before the electroporation. So once again, side effects that need to be addressed for more widespread use. And another system is utilizing lentiviral vectors by using ribonucleoproteins. Now this one is the method that I'm using. There has not been any extensive research on it as much, but I'll be going into more depth on how this method works specifically in a later slide. So my methodology and results. So my results showed, at least when I did the research, that electroporation, as I mentioned, not only is it affordable, but it holds over 90% efficiency in opening up pores in cell membranes. There are concerns, for example, and they had been addressed beforehand about electroporation not being effective. For example, if placed on the human skin surface, we do shock the skin, for example, but how can we be sure it's efficient? That was a long-standing concern. But research has shown that over 90% of cells that they're at least placed onto, they open up effectively, open pores for this transport. And due to this, we can really ensure that with electroporation, we can accomplish almost always the goal that we're looking for, aka transporting effectively these extracellular beings. Now, another thing with electroporation as well, to maximize its 90% efficiency, we can modify how it's used. So, for example, we can modify the magnitudes of power, for example, of these electrodes. For example, a stronger one or a weaker one. We can also change the wavelengths, for example. And with this, it really allows scientists to account for the different types of biomass that these electrodes are putting electricity into. And taking that into account, it's important that we can change how strong the electricity being transported is. And with that, it really does allow electroporation to be used on a wider scale. Results. So Cas9 PLV. This is the method that I was looking into specifically. Now with Cas9 PLV, the way this works is Cas9, a single guide RNA, and just as a side note, the single guide RNA is a specific strand of RNA that tells the Cas9 enzyme that this is a specific strand of DNA that's going to be edited. And that's how the Cas9 enzyme is allowed. The Cas9 enzyme knows that once that specific sequence of DNA is identified, that's where it needs to start the gene editing, which is what makes the sgRNA so important. So with this Cas9 enzyme that actually does the gene editing, the gene cleaving, the Cas9, the sgRNA, and a specific ribonucleoprotein. This ribonucleoprotein allows the Cas9 and single sgRNA to be transported into a singular container of sorts rather than a gene, for example, that's just substituted into the genetic material. Rather, when we can transport this specific CRISPR-Cas9 system into a cell, it allows for the Cas9 to simply begin the gene editing physically in the genome itself, rather than just substituting a specific transgene. And with this specific method, because we have a single guide RNA that allows for greater accuracy in identifying the gene strand of editing, with this Cas9 that actually functions to do this editing, And with the ribonucleoprotein that allows for easier entering into cells without triggering immune system reactions, it demonstrates humongous degrees of safety. There's much lower risk of causing mutations. There's also much less risk of causing immune system reactions. And it's really important that this specific method has addressed these safety concerns, which is what makes it so efficient. When you combine these two methods, though, one can really achieve efficiency in both transport and accuracy. And that's what I did specifically. So when I combine these two methods, the way that this method works specifically is that, let's say on a human, for example, we implement the electroporation, or applying those electrodes onto the human skin, on the human cell, on the human skin surface. And when the human skin surface is shocked, for example, it allows those pores that we're targeting specifically to open up on this area that we're targeting. After which, we inject this Cas9 PLV, or the Cas9 protein lentiviral vector, which combines the ribonucleoprotein, the Cas9, and the single guide RNA. That specific container is transported into those cells. And with that transport, because the electroporation allows for the greater opening of pores, it's so much easier for the specific container to just enter the cell, despite some concerns of it being too big for the cell membrane pores. Because of the electroporation, it opens it up a lot wider, and then it just enters through without triggering the immune system. And then it allows for a safe method without triggering any side effects from the body, and it's a lot more affordable because the electroporation is so widely used. Hence, combining those two methods of efficiency in transport and accuracy. So, my conclusion, it addresses each of these specific research questions. So, HIV, it infects the immune system's CD4 plus T cells, as mentioned before, through the CD4 receptor, the CXCR4 co-receptor, or a CCR5 co-receptor. And using those receptors, HIV is able to inject CD4 plus T cells. Now, how can we prevent HIV from binding to the cells? And as mentioned before, that's where the gene editing technology comes into play here. As mentioned on the last slide, when I explained how we could effectively transport and efficiently do it without as much cost and as much risk, for example, we transport the ribonucleoprotein, the single guide RNA, and the Cas9 enzyme into the nucleus of cells, for example. And when editing the human genome, for example, the sgRNA would have been modified to target that specific gene that expresses the CD4 receptor, or the CCR5 co-receptor, or the CXCR4 co-receptor, depending on the specific sgRNA. And when it's specifically coded that way for the sgRNA to know that this is the DNA segment that's being targeted, when it enters the cell, as mentioned before, the Cas9 enzyme has received those instructions from the sgRNA to keep on going along the genome until that specific gene strand is detected, and then it commences that gene editing. So, for example, if the CXCR4 co-receptor or the CCR5 co-receptor, that specific strand of DNA that expresses that specific receptor or the formation of that on cells, if it's terminated, what happens is those specific CXCR4 co-receptors, or the CCR5 co-receptor, or the CD4 receptor, the expression for that is gone. And after it's gone, there's no more expression for it, and then the receptor, over time, then the cell membrane sort of accounts for that, and it becomes just almost a normal part of the surface. So without the specific receptors, HIV cannot bind. And it's been seen in real life as well. For example, there's a specific segment of the Caucasian population with a mutation called the CCR5 delta 32 mutation. And what this mutation does is it's mutated the CCR5 expression for those people. And what happened is, because that co-receptor is simply just not expressed, HIV was simply just not able to bind to it. And its success was seen through a transfusion of stem cells from those specific mutated people into an HIV patient. And with those specific stem cells, that HIV patient no longer had HIV, undetectable levels, and they were declared as officially cured. And they were known as the Berlin patient, simply just how groundbreaking this was. Because with that mutation, the CD4 plus T cells no longer expressed that specific CCR5 co-receptor. And because of that, HIV was not able to infect any further. And because HIV is not a hardy virus by any means, it dies out without a host. HIV died out, and that patient was declared HIV free. Would this cause complete fortification from HIV? Physically, it should, because without the receptor, it simply cannot infect. And it's important that the CD4 receptor and one of the co-receptors are properly removed, because that way, then HIV cannot infect. Now, there are some drawbacks to removing these receptors, as those receptors, they do serve as some specific communication receptors between T cells. That is a specific drawback, though. Some methods of transporting the gene editing technology, as I mentioned before, we would be using the electroporation on the skin cells, for example, of a human, so that way those pores open up and we can inject the ribonucleoprotein, the sgRNA, and the Cas9 into our specific target CD4 plus T cells. Now, a question as well is, where can we target these CD4 plus T cells? As simply injecting into one's foot, for example, is not going to exactly target a wider range of CD4 plus T cells. And this is where age groups come into play. So as a child, for example, when one's thymus is in development, the thymus is a specific organ that's right around between our lungs, for example. The thymus is an organ where the CD4 plus T cells mature in childhood. And when these CD4 plus T cells are maturing, there's a huge concentration of them there because they're maturing for when we're an adult, when we need those CD4 plus T cells. And the thymus is no longer, as it's not producing those CD4 plus T cells. So within children, for example, if we were to implement the specific method of transportation, it would be essential that it's injected into the thymus, because that's where a huge area of those CD4 plus T cells are present. And if injected there, it really allows for those maturing T cells to never develop those receptors. And without those receptors, HIV simply cannot bind to those T cells, therefore protecting them from the virus. Now, for adults, for example, who may already have the virus, the issue is with adults, by the time puberty strikes, the thymus begins turning from an organ into just basically fat. It starts becoming a lump of fat, basically. And in adults, it's completely fat. And the issue is that you can't just inject our concoction into the thymus, because there are little to no maturing T cells now in that area. But in adults, there are specific areas and specific locations in the human body where there are huge concentrations. For example, lymph nodes and the spleen, for example, there are huge concentrations of CD4 plus T cells in those areas of the body. And if injected there, it really does allow for efficient gene editing in more T cells if injected there. Additionally, with the gut, a gut is an incredibly prevalent area. Around 30 to 50% of a human's T cells are present in the gut. And because of that huge probably like a colony almost of T cells present there, HIV really takes advantage of that. And in fact, there's a huge reservoir in HIV patients in the gut specifically. It's just a huge reservoir of HIV because of that huge colony of CD4 plus T cells in the gut. So if injected there specifically, it can really help in protecting non-HIV infected adults from HIV, because HIV no longer can utilize the gut as a colony. While currently infected HIV adults, if infected there, HIV will not be able to utilize that specific part of the body for future reproduction, and it will heavily impact how many HIV cells are present in the body, as mentioned before, HIV is not a very long-living virus. It really requires a consistently helping host cell to help its replication go forward. Now, limitations. So, as mentioned before, electroporation does cause side effects, for example, to the human, because it can cause rashes, for example. The electric shocks can cause swelling, in some severe cases, skin edema. And additionally, it can really impact the muscle mass of humans, because if the electric shock gets to the muscles, which it has to really get to those cells that are being targeted, it can reduce muscle strength and the muscle mass. And it does take time for them to really regenerate. And that is definitely a valid side effect that needs to be addressed if utilized in the future. Another one is costs. Now, while electroporation is very efficient and very low cost, utilizing the specific lentiviral vector that contains the Cas9, that contains the sgRNA, that contains the ribonucleoprotein, it really is difficult to really bring those costs down. And if we are to implement this in low-income areas where HIV is so devastating, it's important that this specific solution becomes a lot more affordable. So that way, like how ART has been implemented those areas, we can really implement this treatment in such HIV devastated areas, so that way we can really get the majority of HIV patients cured of this disease. So I'm going to be talking about my research experience with the Gifted Gabber program. So my mentorship, I was mentored by Professor Virgil and Professor Kate. Professor Virgil, I would really like to just mention how Professor Virgil was incredibly important in helping me format my paper. Professor Virgil really did present a lot of presentations on some of the new APA format edits, for example, how we properly can cite our resources, how, for example, images, for example, how can we really take, if we are to really get some images from articles, how can we specifically have those images being properly cited so that way it's up to date. And it was really hard for me to actually find them online, some of these new edits, so I'm really thankful to Professor Virgil for helping me format my paper. Professor Virgil is also incredibly important for me for citing my resources. I had a whole list of them, at least 2 or 3 pages, maybe more. And Professor Virgil is really important in helping me format them, put in alphabetical order, so that way it's properly formatted according to JSR guidelines. So I'm really thankful to Professor Virgil for that. And of course, lastly, Professor also was incredibly important in providing and explaining some of these past research examples. So Professor Virgil did show us examples from some past students, and those examples were really important, helping me figure out some of the better ways that I could format my paper, what kind of information I might want to put in my paper, because sometimes, by just saying that, oh, this is what the introduction has, this is what the methodology has, it can be sometimes hard to figure out like what kind of examples you might want to put into these specific paragraphs until you really do look at past papers and figure out how others did it and how maybe you can take inspiration from them, make it your own that way. Next, I really do want to thank Professor Kate. Professor Kate, she really gave us lots of background information on disease modeling. For example, the history of it. We started all the way from penicillin, for example. And starting from there, we really did dive deep into some of these treatments, such as cardiovascular disease. We even touched a bit on gene editing, for example, which is exactly what motivated this topic, because honestly, without that sort of topic like involvement and going to each one, it would have been really hard for me to figure out what kind of topic I really would want to do. And so I'm really thankful that when we really went through each topic in detail, it gave me that idea of what I'm passionate about to research for this specific adventure. Professor Kate was also really helpful in working with me out of our weekly Zoom meetings, to really develop an idea for an effective method of injection. So for example, Professor Kate did propose how we could utilize the thymus in as a proper area of injection. So that way, for example, in children, we're able to really target some of those CD4 plus T cells and prevent the expression of those receptors, so that way HIV just cannot infect those T cells when they're mature. And really, as well, she was important in reviewing my paper. She gave me feedback on how I could improve it, make it more formal, more adhering to research standards, and I'm really thankful for that. Next, I'd like to thank the Gifted Gabber program. The program really did give me insight on how to write papers in the future, because in university, I do want to commit to more research and labs. And with this experience, it really did give me the knowledge on how I can properly write papers in the future, as I'm really interested in genetics as well. So this sort of topic, this sort of program, it really is going to give me, and it has given me, a lot of insight on how I can really write these papers, so that way when I work with professors in universities in the future, I have that base developed, and that way I can really work efficiently with these professors to further this research that I'm just doing right now. Research papers, to be abrupt here, they can be really daunting to read at first, for sure, to a first timer. I remember the first time when I read that research paper on the HIV one, for example, it took me almost the entire day to just really figure out what was going on in the paper. But as I read more research papers, I was able to really understand how to get that valuable information from papers. And without the program, I don't think it would have been possible for me to really gain that knowledge, that expertise, and really understanding research papers without the program. And lastly, the Gifted Gabber program was really important in helping me realize my passion for genetics. I learned genetics a few years ago in AP Bio, for example, or in Honors Bio, and I did like it, as I did like other biology topics. But really, with this experience, with Professor Kate's help and the Gifted Gabber program, it really did help me realize how much I enjoyed studying about genetics. And it's also why I've selected that as my major for college applications. And I'd really like to thank the program for helping me realize the passion of mine, as it would have been a lot more difficult. I might not even have figured it out without the program. One thrilling aspect about the research that I had specifically was just finding out how much research, how many advancements are being made in gene editing every day. Because only a few years ago, like 5-6 years ago, transgenes were really the main method, even though they were not as safe, not as efficient. But now fast forward 5 years later, we're looking into methods to transport the Cas9 enzyme directly into cells, which is so much more efficient, so much more safe for people. And it really just thrilled me to know that maybe 5 years in the future, we could have something that's completely unimaginable for today's standards. So it's really exciting to think about. But yeah, I'd like to thank you guys so much for watching my presentation. And yeah, I'll open the floor.
You can stop sharing. Yep. Mm-hmm. Okay, thank you, Professor Bhatia. Great job. Yeah, so Taksh is really amazing. He likes to do a lot of things very deeply, and we really enjoyed your presentation, Taksh. And we're looking forward to a lot of wonderful contribution of yours. The first being you come back to Gifted Gabber as an intern for our next session and guide kids into that. Okay, so with that, I'll let Professor Kate ask you a question.
First of all, it was really helpful that you said that, you know, reading your first paper took you like all day long, because it's a skill that you develop. But the one thing I wanted to ask you, so we want to, because the electroporation stuff is newer since the last time we talked, what would you package the CRISPR and then the Cas9 enzyme and your guide RNA, what would you package that in to get it into the cell? Have you thought about that?
So there have been past research done, for example, on utilizing a glycoprotein. And the specific glycoprotein, when tests had been done on this specifically, what the glycoprotein really did show is that when it entered those cells, it didn't trigger any reactions by the cell, for example. Again, it simply passed through without triggering anything. And I think it's perhaps also because you can find glycoproteins on the cell membrane surface, for example, and that sort of similarity, it must have been important in really allowing it to pass without triggering anything. And that glycoprotein, it was also very durable for the specific contents being transported into the cell. And I think that's really important, that we can really ensure that these contents can be transported into the human body through the extracellular matrix, and then the cytoplasm, then into the nucleus. So it's really important that this container, this glycoprotein container, it's durable and it doesn't trigger any reaction, so that way it can achieve its intended destination. So yeah.
Great. Thank you. You did a wonderful job.
Yeah, excellent, Taksh. Thank you.